Welcome to all students. Today we are going to discuss a very important topic related to the chapter number 2 of the NCERT, that is the sexual reproduction in the flowering plants. And today we are going to discuss the development of embryo in dicots. It is very important one, and you must know this embryonic development in a proper sequence. So have a look. We are actually studying the development of embryo in a special dicot plant, which is a typical angiosperm known as the Capsula brusa pastoris, which is belonging to a family that is known as the Cruciferae. And what the embryonic development we are studying, which is the most common type found in capsula, is called as the crucifer type of the embryonic development, or we can say it as Ona grade type of the embryonic development. Okay. Now, as we know very well that the double fertilization and triple fusion result in the formation of a diploid zygote, that we know very well. And in the case of the angiosperm, from this zygote we will be developing a embryo, and the embryo which will be formed will be also a diploid embryo. Okay. Now, in the case of the angiosperm, the zygote which is formed by the fertilization, that is, which is formed by the fusion of a male gamete with the egg cell, then a diploid zygote is formed. This diploid zygote undergoes a period of rest and forms a layer around it during that rest period. And when the zygote is having a layer around it, that zygote is called as the oospore. Okay. Now, this zygote or the oospore will first of all divides by a transverse division, right? By a transverse division like this type, and you can see the diagram number B that two cells are formed. Okay, due to the transverse division in the zygote, what happens? Two cells are formed. One is known as the terminal cell, which is also called as the apical cell, or it is also called as the embryonal cell. And the another one is known as the basal cell or the suspensor cell. Okay. Now, always remember, this terminal cell or the apical cell or the embryonal cell is always present towards the chalazal end. You know now that the entire embryonic development is going on in the ovule, and the ovule has two ends. One is known as the micropyle and another is known as the chalazal end. So what I want to say, that this terminal cell or the apical cell, right, or the embryonal cell is present towards the chalazal end. Okay. And the basal cell or the suspensor cell is towards the micropylar end. Fine. Now always remember, the basal cell is called as the suspensor cell because it will form the suspensor. And the terminal cell is also called as the embryonal cell. Why? Because this cell will form the embryo. So a embryo forming cell is called as the embryonal cell, and a suspensor forming cell is called as the suspensor cell. Okay. So like this type, two cells are formed. Now what happens in the next step? Now you have to concentrate on this diagram number C. Now what happens? That this terminal cell or the apical cell or the embryonal cell found towards the chalaza divides vertically. Now vertical division is like this type. This is the vertical division, and like this type, this is the transverse division. So what I want to say, that this apical cell or the embryonal cell divides vertically, right? So when it will be dividing vertically, so here you can see there is one terminal cell. When it will divide longitudinally or the vertically, so two cells are formed. So these are the two embryonal cells. Fine. These are the two embryonal cell, right? At the same time, what happens? That this basal cell, right, which was towards the micropylar region, also divides. But now this cell divides transversely, like this type, not like this type. It will divide like this type, transversely. So two cells are formed. Now these two cells are called as what? The suspensor cells. Okay. So now we can say it as that the embryonal cell has undergone division. It has divided vertically to form two embryonal cell, and the basal cell has divided transversely to form two suspensor cell. So altogether now there are 4 cells, and this 4 cell structure, you can see that it is representing a T-shaped structure, right? It is representing a T-shaped structure, somewhat similar to the alphabet T, and hence it is called as the T-shaped structure. Now what happens? Now you have to focus on the diagram number D. Now what happens? This embryonal cell, right? How many embryonal cells are there? 2 embryonal cells. These embryonal cell will now divide, okay? Will now divide again, right? And will form how many cells? Now it will divide transversely and will form 4 embryonal cells. So here there are 2 embryonal cells. Here we can see that how many embryonal cells are formed? 4 embryonal cells are formed, because mitotic division is undergoing, and the 2 embryonal cell will be converted into 4 embryonal cell. So here we can say that these 4 embryonal cell, right? The embryo with 4 embryonal cell is called as a quadrant stage. Quadrant word is used for 4. So I can say that this is a 4-celled embryo, right? Which is representing the quadrant stage. At the same time, now this suspensor cell, these 2 suspensor cell will undergo many transverse division, and due to the transverse division, due to this, what happens in suspensor cell? Which divisions occur? Transverse division occur, right? And a structure is formed from these 2 suspensor cell, and this structure is called as a suspensor. And this 6 to 10 cell structure, suspensor, basically now the question arises that why this suspensor is formed. So actually the suspensor is attached to the embryo. So this suspensor pushes the developing embryo deep inside the endosperm to provide nutrition to it. Okay, once again I am saying the suspensor pushes the developing embryo deep inside the endosperm. We know very well that inside the embryo sac where the fertilization has occurred, at that place endosperm was also formed due to the double fertilization, and endosperm is a nutritive tissue. So this suspensor will push the developing embryo deep inside the endosperm to provide nutrition to it. Okay, and remember the suspensor, right? The suspensor cell, this suspensor cell, okay, which is away from the embryo, right? This cell, or we can say that the terminal cell of the suspensor, which is away from the embryo, swells and it forms the hostorial cell. Means the hostorial cell is always towards the micropyle. You can see this diagram. This is the diagram of the ovule, and you can see that just near the micropyle is the swollen cell of the suspensor, and this swollen cell of the suspensor is called as the hostorial cell to procure the nutrition, right, in the form of the endosperm, right? So this is the hostorial cell, and always remember the cell present near the embryo, right? So this is the suspensor. This is called as the hostorial cell, which was towards the micropyle, and the cell which is present near the embryo is called as hypophysis. This cell will be called as what? The hypophysis. I have mentioned here. This is the hypophysis. Fine. So this is hypophysis and this is hostorial cell, and this entire structure is the suspensor. Fine. Now what happens that now forget about the suspensor. Now you have to concentrate on these 4 embryonal cell, which, or we can say it is the embryo which is in the quadrant stage. Now what happens in the next stage? You just focus on the diagram number E. Now these embryonal cell, right, which are now 4 in number, will again divide by mitosis. And we know very well that mitotic is a division which doubles the cell numbers. So the 4 embryonal cell now will be converted into 8 cells. So this will be called as 8 cell embryo. See here, 1, 2, 3, 4, 5, 6, 7 and 8. So this is a 8 cell embryo, and for the 8 cell embryo we use the term octant stage. This was known as the quadrant stage. This is called as what? The octant stage. Okay, the cell of the suspensor present just near these embryonal cell is called as the hypophysis. Now the question arises, what is the role of the hypophysis cell? So this hypophysis cell, right, will form the root cap. You know very well that root is covered by root cap. Root is covered by root cap. So hypophysis forms the root cap. Okay. Remember, hypophysis never forms the root. It is forming the root cap which is present at the tip of the root. Okay. So hypophysis forms the root cap. Now this is a 8-celled embryo. Now out of the 8 cells, the four cells which are present towards the chalaza, the 4 cells 1, 2, 3 and 4, there are 1, 2, 3 and 4, there are total 8 cells in the octant stage embryo. So the above 4 cells which are present towards the chalaza, right, the above 4 cells which are present towards the chalaza, right, are called as the epibasal cells, and the 4 cells, the 4 below, hypo means below, epi means above. So the 4 hypobasal cell, these 4 hypobasal cells, okay, so the cells present below the epibasal cell are called as the hypobasal cell, right. So these epibasal cell will form 3 things that are very, very important one. The 4 epibasal cell which are present towards the chalaza will form the 2 cotyledones. We know very well that in the dicots there are 2 cotyledones. So will form 2 cotyledones, a epicotyl and a plumule. Okay, what happens? That 3 structures are formed: 2 cotyledon, 1 epicotyl and 1 plumule. So if somebody asks you that the cotyledones and the epicotyl and the plumules are made by, so they are always formed by the epibasal cells of an octant stage embryo which are present towards the chalaza line. Now what about the hypobasal cell? So the hypobasal cells form the 2 things, radical and hypocotyl. Always remember, it forms radical and hypocotyl. So again I am repeating, epibasal cell will form the 2 cotyledones, epicotyl and plumule. And what about the hypobasal cell? Hypobasal cell will form the radical and the hypocotyl. Okay, so this is an octant stage embryo representing 4 epibasal cell and 4 that is known as hypobasal cell, and hypophysis, which is a cell of the suspensor, right, will form the root cap. Now this was the diagram number E. Now just focus on this diagram number F, okay, and which is very important. Now this is a globular embryo. This stage is called as the globular embryo stage, and again these 8 cells, right, that is the octant stage embryo, again will divide by mitosis, and we know very well that in the mitosis the chromosome number will be doubled. So the 8 cell will form how many cells? That is known as the 16 cells. Now always remember that 8 cells are found in the outer layer and 8 cells are found in the inner layer. Okay, how many cells are there? 8 cells are found in the outer area and 8 in the inner area. So this is a 16-celled embryo. A 16-celled embryo is called as a globular embryo. Okay, and this is the same thing. This is the suspensor. This is the hypophysis cell, and this is the ostodial cell. Fine. So this is a globular embryo. Now soon what happens, that the divisions are regularly going on. The mitotic division regularly goes on, and this globular embryo, which was a 16-celled embryo, converts into a heart-shaped embryo. Now you have to be peculiar about the sequence of the embryo. Okay, now first we have talked about the globular embryo. Now I am talking about the heart-shaped embryo. Okay, and remember that the divisions which are going on in the globular embryo will form a heart-shaped embryo, and all the cells of the heart-shaped embryo are meristematic, right? So we can see that a heart-shaped structure is formed, and you can see that 3 layers are formed here. How many layers are formed here? 3 layers are formed. We study in the plant anatomy in the class 11th that what happens, that if these cells are meristematic, so during the embryonic stages these meristematic cells of the heart-shaped embryo will form 3 layers. Okay, will form 3 layers. The outer one is known as the dermatogen, the middle one is called as the periplum, and the innermost is called as the plerome. As we have studied in the plant anatomy, that dermatogen will form the epidermis. Dermatogen is the outermost layer and it will form the epidermis. Fine. And periplum, which is the middle one, will form the cortex and the pleurum will form the vascular cylinder, that is the xylem and the phloem. Okay, so 3 layers are also differentiated here, dermatogen, peribulum and pleurum, and you can see that 2 lobes are formed which are actually representing the cotyledon, okay, and in between this you can see that a plumule is also there. Fine. Now at the last you can focus on this diagram number H. This diagram is actually representing the mature embryo. This diagram is representing the mature embryo, or it is also called as the torpedo stage, Here you can see that this mature embryo is little bit curved. Okay, this embryo is little bit curved, right? And you can see that the hosturial cell is present near the micropyle. Just behind the hosturial cell are the cells of the suspensor, right? And the cell of the suspensor which was present near the embryo, which was known as hypophysis, has formed the root cap, okay? So the hypophysis cell has formed the root cap, right? This is a mature embryo in which you can see that the 2 cotyledons are there. Here you can see the plumule. Here is the cylindrical structure called as the hypocotyle, right? This hypocotyle forms a radicle, and radicle will form the root tip, and root tip is covered by the root cap. So this mature embryo is basically found in this ovule, right? As we know very well that the ovule here is in the case of the capsula is called as the campylotropous ovule, and it is a curved ovule, and in this curved ovule, right, a curved embryo is formed which is known as the torpedo or the mature embryo. Okay, now when we are talking about the embryo, we must know certain terms. Okay, say for the very first is the embryonal axis. Okay, embryonal axis is also called as the tigellum. And it is the axis present between plumule and radicle. Always remember the axis which is present. See, plumule give rise to shoot and radicle give rise to root. So the axis which is present, between, say for if this is the axis, so this is the embryonal axis, the axis which is present between the plumule and the radicle, the axis which is present between the plumule and the radicle is called as the embryonal axis, which is also called as the tigellum. Now there were few terms here. Epicotyle, what is epicotyle, right? Cotyledon, plumule, radicle, hypocotyle, you must know. Now first of all you must know that what is epicotyle. Epi means above, hypo means below. So epicotyle is present above the level of cotyledons and hypocotyle is present below the level of cotyledons. So the portion of embryonal axis above the level of cotyledons, remember the portion of embryonal axis above the level of cotyledons is called as the epicotyle, and always it terminates into plumule, or stem tip, right? And another one is known as the hypocotyle. Hypo means below the cotyledons. So the portion of the embryonal axis, below the level of cotyledons, is called as the hypocotyle, and hypocotyle terminates into a cylindrical structure which is called as the radicle or the root tip, and root tip is covered by the root cap, and that root cap is formed by the hypophysis of the suspensor cell. Okay, so these are the terminologies. You must know that what is embryonal axis, what is epicotyle, and what is hypocotyle. Okay, and what is plumule and radicle. So always remember, in the case of the dicots, 2 cotyledons are formed. That's why they are called as the dicots. So this was the total embryonic development which occur in the typical angiosperm, typical dicot known as the capsula brusa pastoris, and this type of the embryonic development which I have represented here is also called as the crucifer type of the development or the onacrate type of the development. So you must study the development of the embryo in these series of steps just as what I have shown you. Okay, so there must not be confusion, because this is the micropylar end, and towards the micropylar end the suspensor is present, because of which the suspensor will absorb the nutrition in the form of the endosperm and will provide the nutrition to this developing embryo. Fine. These are the 2 These are the two cotyledons. This is the plumule. This is the cylindrical structure known as the hypocotyl. Hypocotyl give rise to radicle. Radicle give rise to root, and root is covered by the root cap. Now this entire embryo, what we have studied, this embryonic development is going on in the ovule. But we know very well that after the embryo is formed and the fertilization is over, the ovule converts into seed. This ovule converts into seed. Okay, so where is this embryo? Somebody asks you, where is the embryo? So we will say embryo is present in the seed. Why? Because after fertilization, ovule is converted into seed. So where is this? If ovule is converted into seed, and earlier the embryo was actually present in the ovule. So if ovule is converted into seed, so where is the embryo? Embryo is present inside the seed. So always remember, if somebody asks you that what is embryo? So embryo is a tiny plant. It is a tiny plant which is present in the seed. When you will be sowing the seed in the soil, and if you will be irrigating the soil, what will happen? A young seedling will come out. So that young seedling which is coming out from the seed, it is nothing, it is the embryo only. So the tiny plant present in the seed is called as embryo. So remember that after fertilization, right, ovule is converted into seed, and inside the seed what is present? The embryo is present. And where is this ovule present? The ovule was present in the ovary. So after fertilization, ovary converts into fruit. Ovary converts into fruit. So I can say that what happens? Ovule converts into seed and ovary converts into fruit. And there are certain changes also which happens, that the integuments, right, which were the protective coverings found around the ovule. So when the ovule converts into seed, then the integuments of the ovule will form the testa of the seed. Now what is testa? So testa is nothing, it is the seed coat. Testa is the seed coat. There are two testas, outer seed coat and inner seed coat. Now integuments were also two in a bitegmic ovule. So outer integument will form outer seed coat, inner integument will form inner seed coat. Okay, so the outer one is called as the testa and the inner one is known as the tegument. Fine. So this was the embryonic development. Hope so you have liked this video. We will be coming soon with few more videos, and if you want to take the screenshot of this video, you can take so that you can maintain the notes. Thank you.